see the...
Yeah, that's working. Yes, Irene, please go ahead. We have participants joining, but you won't be able to see them because it's a webinar mode. So whenever you're ready, it's 10, we can start. Enjoy the webinar and welcome everyone. Great to see you all.
So I think we should wait something about 2 minutes, if it's okay with you. Thank you for coming in this fourth webinar. We will start in about 2 minutes, okay? Waiting for more members to join. Okay, good morning, everyone. I think we are ready to start. So, salinization is a major driver on land degradation, affecting almost 800 million hectares worldwide. Sustained Coast Action, funded by European Cooperation in Science and Technology, aims to build a global, transdisciplinary network of research and stakeholders focused on salinity in relation to sustainability and food security. Advanced understanding of plant soil responses to salinity develop improved management practices and highlight the value of salt-affected landscapes, promoting knowledge exchange and more resilient, sustainable use of salt-affected lands. Sustain is organizing a series of webinars seeking to build a shared knowledge base to improve water and soil management and enhance crop production on salt-affected lands. So, good morning again, and I would like to welcome you on the fourth Sustain webinar with the title of Navigating Salinity Stress in Rice, Interactions with Far-red light and the Advantage of Lateral Growth. So we have today with us two speakers, and I would like to welcome on the floor the first one, Parvinder Kalon, Rice Breeding Innovations, and from the IRRI, the Philippines, and Laboratory of Plant Physiology of the Netherlands, with the topic of interactions between salinity and supplemental far-red light responses in rice. Parvinder, the floor is yours.
Thank you so much, Irini, for the kind introduction, and good morning from my side. So I will start sharing the screen. All right. So a good morning from my side to everyone present here. And I would like to first of all thank the organizers for giving me the opportunity to talk today in the webinar. And today I will be talking about our work on rice and interaction between salinity and supplemental far-red light responses. And I have split my talk today in two parts. The first part, I would be talking what we performed or worked on at Wageningen University and research in the Netherlands. And it majorly focuses on early seedling development and how the rice responds to these two stimulus. And the second part would be more like a teaser, which I would be presenting our field data or the field trial, which we are currently performing at International Rice Research Institute in the Philippines. So let's get started. So as many of you in the audience must be aware that rice is one of the plant species which is quite sensitive to soil salinity. And on the screen you see, on the right-hand side, this is the field in north of India, which is used to cultivate rice. And the crust of this white crust on the top layer you can observe is salt. And if you grow rice or plants in such a field, the plants develop defective phenotype, which is documented over here. You can see that the plants develop chlorotic lesions on the leaves. And ultimately, this in the field leads to reduced tillering and spikelet sterility, as well as patchy growth in the field, leading to low yield. So it becomes a major concern for rice cultivation at large scale as well as at small scale farmers. And simultaneously, what we observe in rice cultivation, there is a major switch. So the major switch being from the traditional submerged paddy field shown over here towards direct seeded rice cultivation, which is shown on the right-hand side of your screen. So just to walk you through, submerged paddy field is that before the rice is cultivated, farmers submerge it with a lot of water and then individually sow the plants, which are already grown in the greenhouse for 10 to 20 days. And doing so, you can have an optimum density in the field, and it's very easy to control the weed in this kind of field, but it is very costly and labor intense, as you can see also in the picture that you need individual labor to sow these seeds or these seedlings. And it is also very unsustainable because of high methane emission from a submerged paddy. So there is an obvious switch throughout the world towards doing or practicing direct seed rice cultivation. And that in small scale farmer field looks like just directly spreading the seeds in the field. And why so? Because it's very efficient and it's sustainable and it's an economically viable option when we look into the small scale farming. But it comes with a cost. And the cost being that when you do manual spreading, there is an uneven planting. And now we are switching from submerged paddy field to just a wet or uniformly wet field. Then we have a lot of weed proliferation in that case. And what happens when there is a weed proliferation or uneven planting? The plants experience higher far-red reflectance. And this, in turn, plants perceive it as a shade cue. And further, this shade cues can come from its own shoot architecture, from the uneven planting, and as well as the weed which is growing in the neighboring side. This ultimately leads to a poor crop strand. And what we know about these two stimulus or stressors in plants, that how plants respond to these two, which was shown in Arabidopsis from work from our group by different colleagues, that when you see in Arabidopsis, the plants, when they are grown in supplemental far red, which is a cue for shade, they show petiole elongation, which is a very typical shade avoidance response in plants. And then when we expose these plants to mild salinity of 25 millimolar, this petiole elongation is suppressed or diminished. And what the colleagues were able to show in this work, that this suppression is ABA dependent. So in a schematic here, you can see when the plants are, let's say, treated with sodium chloride, salt in this case, the plants activate an ABA pathway, which further leads to halting of a brasinosteroid pathway, ultimately leading to suppression of elongation, auxin signaling elongation, which was initiated by the shade avoidance response. But when we talk about rice, so when we look in different plant species, the typical shade avoidance response is a hypernasty or like upward growth, which can be seen over here. There are different plant species, Arabidopsis, tomato, that when the plants are exposed to a far red light, a cue for shade, the plants are always growing upward as compared to plants when they are not exposed to far red light. And in rice, we document this upward treatment or upward response in form of doing the culm height documentation, which is indicated over here. And when we see in rice, the plants, when they are grown in white light, and then when you have the plants which are grown in supplemental far red, surprisingly show a very contrasting phenotype when compared to different plant species. It's not an upward movement, or let's say upward growth, but it shows a stunted phenotype. And this work was led by one of our colleague, Martina Huber, and where we were able to show this response is in rice variety dependent. So on this graph, if you notice, it's different varieties of rice, and on x-axis you see culm height, and there are varieties indeed which show this upward movement or elongation when compared to plants grown in control condition or white light. But then there are varieties which show a opposite trend, so they don't show an elongation of culm, but they show a stunted phenotype. So this kind of made us wonder, first of all, how rice in particular responds to the shade cue, in this case, supplemental far red. And also additionally, if there are any mechanism we can underline or pinpoint to, which has an interaction between supplemental far red and salinity signaling in rice responses similar to what we observed in Arabidopsis. So to do so, we set up a standard physiological experiment where we looked into growth and development of rice, in this case, Nippon Bare, which is one of the variety which is traditionally or very well used in different labs. And we looked into in a hydroponic solution to start with to get a very cleaner system. And what we did, that we take plants in the first, in a radical stage, and then we treat the plants for 14 days with either salinity, a very mild salinity at 25 millimolar, or at supplemental far red, or we did also combination treatment, salinity plus supplemental far red, and then we looked into the growth phenotypes. And now for a couple of slides, you will see this color coding. If you see the blue bar plots or box plots, then it means that the plants were treated, they are control condition. And the plants, if you see a yellow bar plot or box plot, then it's plants which were treated with mild salinity. And if a brick red, this would be supplemental far red. And the orange plot would show the combination treatment. And what we were able to see in the culm height in Nippon Bare at 14 days treatment experiment, that when we document time-wise or to a time series of the culm height, that we can see starting from day 5, the suppression of culm height upon different stress stimulus application. And at day 13, it becomes very evident when you see the plants which were grown in control condition and the plants which were grown in mild salinity condition were significantly suppressed or stunted. And this suppression was even more prominent when the plants were treated with supplemental far red, and the combination treatment even went smaller. And when you look at 14 days, a schematic or like just a representative picture, the plants show very distinctive phenotype, plants which were in white light, or plants which were in mild salinity, plants which were exposed to supplemental far red, and plants which were exposed to combination treatment. And when we look at the biomass of root and shoot of these plants exposed for 14 days to these treatments, when we look at mild salinity treatment, shoot was able to show significant reduction when compared to control treatment, but root didn't show any significant difference. And surprisingly, when plants were exposed to supplemental far red, we saw an increase in shoot and root drive rate. And interestingly, when there was a combination treatment, so the presence of salinity and supplemental far red, this increase was suppressed, which we observed similar to Arabidopsis. And then we wondered, like, what kind of, for instance, molecular mechanisms are upregulated or downregulated in this interaction. So to do so, we took a transcriptomic profiling approach where we grow the plants for 5 days in control condition. Then we treat the plants with these 4 different treatments. For 1 hour and 3 hours, we separate the plant's shoot part and the root part. And then we went for bulk transcriptomics. And when we looked at differentially expressed gene, what we were able to see was that upon salinity treatment, so now I would be focusing mostly the data on shoot because of the time constraint, but I will give you a little bit teasers throughout my talk about what happens in the root as well. So when we focus on the shoot part at 1 hour at salinity treatment, we were able to see almost no differentially expressed genes. And this was kind of expected because 25 millimolar is a very mild salinity. It's pretty much on the edge for early treatments to see any differences. And the differentially expressed genes numbers were higher when the plants were exposed to supplemental far red, shown over here at 1 hour and shown over here at 3 hours. So there was a progression. Upon 3 hours, we find more DEGs. And then most striking part over here was the Venn diagram over here, which shows plants which were treated to combination treatment show unique or reveals unique differentially expressed genes, which were not present alone in the salt treatment or alone in the far red treatment. And this was the similar case in root as well. And when we look at these genes in the list, there was no surprise that we find a lot of DEGs which shows genes from phytohormone pathways. And that kind of was expected, but also we know from Arabidopsis that ABA was the key player in this interaction response, and particularly in salinity, we know that in rice, ABA plays a major role. So then we started looking at the endogenous level of this ABA in the shoots after treatment with, so we took approach where we grow the plants for 5 days in control condition, and then similar to transcriptomic approach, then we treat the plants for 3 hours and 6 hours, and then we take the shoots and roots, and then we look into the endogenous level of APA. And what we were able to see that salinity had no significant differences when compared to control. But when we looked at supplemental far red plus supplemental far red plus salinity, that in both the time points, ABA was significantly increased when compared to plants which were present in the control conditions. And simultaneously from our group work, we found out that when we look at Arabidopsis in ABA mutants, indicated over here, they showed, so we took 2 ABA mutants, ABA3 and SNRK 2.2 and 2.3, and when the plants were, these ABA mutants were treated with sodium chloride of 125 millimolar for 6 hours, were able to show significantly higher level of sodium accumulation in the shoot part, but no difference were observed in the root part. And that kind of made us wonder that if, like now we see that supplemental far red in rice increases an ABA level when compared to control condition. And in Arabidopsis, we know that when the ABA is absent, there is a higher level of sodium in the shoot. So we were wondering that if we see this protective mechanism in the rice or this biomass increase in the rice, that if this is somehow regulated via iron accumulation or iron homeostasis. So to do so, we took an approach of doing an elemental analysis. And today I will be showing you data from chloride. So we looked into sodium, chloride, and potassium, and chloride being one of the key component. And I'm just taking an approach to show chloride because we were able to do a subcellular localization of chloride. And right now we are trying to put efforts on doing the subcellular localization of sodium as well. But the trends at bulk level, we observed similar for sodium and chloride. And that being, in the figure you see the plants, just the phenotypic response, the plant which was exposed to alone salinity, and the plant which was exposed to salinity plus far red. And when we look at the shoot level, chloride level, that they were significantly reduced when there was presence of supplemental far red. So somehow supplemental far red is activating an ion homeostasis level in the shoot part, that it's the bulk level of chloride as well as sodium goes significantly lower. And this difference was not evident in the root parts. And when we wanted to then look at where at subcellular level in the leaves it is present. So to do so, we performed an elemental mapping at X-ray using X-ray microscope at the European Syndrome facility in Grenoble, France. And what we were able to see that when we do a cross section of leaf and the plant which was exposed to alone salinity, and that we were able to see chloride was present throughout the leaf cross section, but there were areas where there was concentrated levels. And then when the plants which were exposed to supplemental far red and salinity, overall the bulk level was lower, but also the localization changed, that you see that the chloride level was much higher in the epidermis of, or like a metabolic inactive part of the leaf as compared to overall throughout the leaf. There was like a bit lesser as compared to when you see in the plants which were treated to salt alone. So this, until now, what we can say is that far red light somehow sensitized the plants to even very mild salinity in early responses. But the main question stays, is it beneficial or detrimental in the long run? At early seedling stage, it does seem like a beneficial approach, but we have no clue how it would perform in the field. And to do so, we performed an experiment in the field in International Rice Research Institute in the Philippines, and to look into the later stages as well, that how the plants perform when they are simultaneously exposed to salinity plus shade cues. And what approach we took over here was that we took 3 different salinity treatments. So one being control, and then mild salinity of electric conductivity 6, and then high salinity, which is electric conductivity of 12 decisiemens per square meter. And then we also took treatment, 3 different density treatment, 1 recommended density, then 1 mid density and 1 high density. And we took 5 different varieties, all of them indica because it's in Philippines, so we cannot grow Nippon Bare there. It's a japonica variety. And in this indica varieties, we took 3, one which is a breeding line, widely used nowadays, one of the elite line. And then we took a variety which was salt tolerant and a variety which has been shown shade tolerant, and 2 varieties which in our lab conditions showed a contrasting phenotype, where IR64 shows the figure I showed you earlier. IR64 shows stunted phenotype to supplemental far red, and Mudgo shows an upward growth or increased column height when plants were exposed to supplemental far red. And then we took a 3 meter square plots, and we did 4 plots each treatment, and then each variety. And today I will just show you. So now the data is from one of the field trials, so we can indicate what it goes through, but then we have to repeat a couple of times, at least 2 more times, to say robustly what we see. But what we see so far, one of the key score we look at is the standard evaluation system scoring, which is a salinity score widely used for rice. That being that if plot gets a score of 1, that means the plants are showing no severe phenotype or there is no leaf symptoms and the whole plot looks very healthy and green and they are highly tolerant variety. And it progressed up to 9, where 9 being the worst, that all the plants died in that salinity treatment in that particular plot. So what we were able to see is that now I'm showing you 2 graphs over here, one for EC6 and on the right hand side is EC12. And then there are 5 varieties and 3 line plots per variety. The first line is green, which is control density. Orange line is mid density and purple line is high density. And we started the treatment at 40 days after sowing. And then we started documenting SES scoring every week. So just keep in mind that if higher the SES score, that plants are performing worse, lower the SES score, plants are performing good. So they are highly tolerant to salinity. And what comes up in different varieties, for instance, at EC6, if we see that there is somehow early stage protection in density. So green line, which is the control density, is always performs worse in all of the varieties as compared to orange plot or purple plot, which are higher density. But then towards once we stop the salt application, somehow plant catches up in the overall level. And it's a mild salinity and rice is shown to be very plastic in shoot responses. So we would expect that. But this is a bit different when we take a very high salinity, which is EC12, that for instance, let's focus on IR6421, one of the moderately tolerant variety to salinity, that the control, there is an early protection in a very high density cropping system that their plants show a bit better performance and lower SES score. And towards the end, even the salt application was stopped, the plants showed higher protection when there was higher density treatment as compared to the control density treatment. And then we also looked at root. So to do so, because root being the main factor over here in response to salinity. To do so, we did soil coring, where we take a 60 centimeter of region between the 2 hills of 2 plants, and then we use a soil borer and take the 60 centimeter soil and separate it in 4 regions. Region A being 1 to 15 centimeter, region B being 15 to 30 centimeters, and C 30 to 45, and D being 45 to 60 centimeters. And what we were able to observe, so today I will show you one of the data for dry biomass, but we also did a couple of other parameters, and major differences we observed in region A and B. So the data will be shown for region B. So what you see over here, region A plot, region B plot, 5 different varieties, and then EC0 control treatment in the first column, then EC6 mild salinity treatment, EC12 high salinity treatment. And what is very evident over here is that 2 of the variety IR64 and IRRI147, one moderately salt tolerant and one salt tolerant, shows to perform better in salinity treatment under higher density. So if you let's focus on this part, which is mild salinity, the first being control treatment, then mid density and high density. The root dry weight was much higher in high density, and in region A as well as in region B. And similarly, the case was for 147. And there was another interesting variety, which is 154, which showed these differences, significant differences in control condition in region A, but these differences were diminishing when the salinity treatment was performed, as you can see over here. So overall, what we can say over here is that IR64 and 147 shows higher root dry weight at higher density, but overall we do see these trends in different varieties. For instance, we don't see that trend of increase in density for macro, which is very susceptible to salinity. And then we do see that trend in 1 to 3, but this is not significantly different. So this would tell a bit more when we repeat a couple of times this field experiment that what actually is. So in this part, what we can say so far is that effect of salinity and density on rice growth is potentially show root-based protection, but only in few varieties. And what we see is that higher density may reduce salinity impact dependent on the variety and the salinity treatment opposed on the field. And yes, with this, I'm towards the end of my talk, and I would like to thank a lot of people which were involved in this experiment. But first of all, a big thanks goes to Krista, Ronald, and Rumiana for giving me the opportunity to work on this exciting project. And also a bunch of thanks goes to IRI, Wageningen, ESRF, and Microbe for funding this project, and many groups which were involved in this project: Roots in Salt Group, Laboratory of Plant Physiology at Wageningen University, Laboratory of Plant Physiology and Laboratory of Phytopathology at IRI, and members at Wageningen and members at IRI who helped me through this big task. And thanks to you guys today for attention.
Many thanks, Parvinder, for this interesting presentation. It was really very interesting for me and our members, for sure. So I will have time for questions at the end of the webinar. And now I would like to ask to the floor Yori Leshem from Tel Hai University and Mikal Galilei Research Institute from Israel on the topic Islands of Hope, the advantage of going sideways during lethal salinity. Yori, the floor is yours.
Thank you very much, Irene. Can you please tell me, let me know if the slide is moving?
Yes, it's moving. Thank you very much,
Okay,
Yori.
great. Thanks a lot. So, hello everyone. I hope you hear me well and you see my slide. My name is Yori Leshem. I'm from Tel Hai University of the Galilee in Northern Israel, and I hope to share with you some of the work we've been doing in my lab. But before starting my talk, I would first like to begin with acknowledgements and thank the organizers of these great webinars for the opportunity to speak here. Thank you very much. I appreciate it a lot. I would like also to give credit to those who actually did the work, mainly my former postdoc, Dr. Vivek Ambasta, a gifted scientist, which is now doing a second postdoc in the USA. And of course, the funding agency that supported us, the Israel Science Foundation. Okay, so, it's a story. It all started a long time ago during my PhD. I don't want to tell you when was it. It was a long time ago. You see my balding head. But I was studying lethal salinity, and I was working on Arabidopsis. And sorry for that, but I believe the results I present here are relevant to this forum. So what we see in front of us is Arabidopsis seedlings and treated for 4 days with 200 millimolars of sodium chloride on the left, which apparently is lethal. It's like 10 times more than the previous concentration or the great talk on rice. So during my studies, I mainly focused on early responses of the roots to this lethal concentration. In this case, we see a reactive measurement of our reactive oxidant species generation immediately after exposure to salt. And as mentioned earlier, this lethal concentration kills the roots. And the commonly held view until recently was that the root dies, the Arabidopsis root dies within couple of hours or 2 to 4 hours, it will be entirely dead. Towards the end of my studies, I said to myself, after all, the root is not uniform. It is composed of various cell types and various cell identities. And I was wondering, is it possible that a certain cell dies quicker, or is it possible that a certain cell persists longer? Unfortunately, um, this question remained unanswered because after completing my PhD, I moved on to California, to UC Davis, where I studied other stuff. I can tell you about this some other time. But then, towards the end of my stay in California, I was lucky to get to know Professor Siobhan Brady, a very important scientist. Back then, she was young faculty. And Siobhan came from the lab of the late Professor Phil Benfey, who developed a series of cell-specific reporter lines in the roots, and which he was very kind to share with me, and I'm very grateful to her for that. So here is these lines, and when I returned back to Israel and opened my own lab, after a while, I managed to get some money, and then Vivek joined me, and together with him, we started working as a side project on this issue. And this question was stuck in my head, and it was remained unanswered. I said, "Wow, let's we have the opportunity. Let's go and try to answer this question." So together with Vivek, we challenged these lines with little salinity concentrations, and then determine the cell's viability, which was indicated by GFP persistence, and it was combined simultaneously with vital staining. Now, if I had more time, I would now make a quick quiz and ask you which cell type you think persisted longer. But I don't have spare time. And actually, I don't know the answer. I don't have a good answer for this question. Because after some time of hard work, Vivek came to me and he said, "Yori, they are all dead." He was working on these lines, are all dead. And this is what he saw. So this is the control, one of the lines. So we see a very nice GFP signal of scarecrow, which indicates the endodermis, a single layer of endodermis and the quiescent center in the bottom. I'll just turn on my laser pointer, and the red propydium iodide signal is located only on the cell's membrane, not inside the cytoplasm. And this is like how a healthy root looks like. But after harsh treatment of 200 millimolar sodium chloride for 48 hours, the GFP signal faded, and the PI, the propydium iodide, penetrated into the cells, actually into the nucleus, indicating cell's mortality everywhere. Everything was dead. So this was kind of disappointing. But then Vivek said, Yori, but the lateral roots remained alive. And this is what he saw. So let's now see the lateral position. So this is a young lateral root budding out of the primary root, of the main root. And the same signals, we see the same scarecrow signal, beautiful signal of scarecrow. The propydium iodide is on the membrane, the periphery, obviously it's very healthy. When we exposed it to, after exposing it to salinity, the main root cells died. You see all these red dots? I have a red dot on the top of red dot. You can't see it, so maybe I should turn off. But you see a lot of red dots all around the dead cells. But in the middle of this image, we see very nice GFP signal of the lateral root, which persisted, and the propydium iodide did not penetrate into the nucleus, indicating that this region is alive. Is still alive after 48 hours of deadly salt. And this is the island of hope I mentioned in the title of my talk. Now this result amazed me for two reasons. First one, it was so unexpected. As mentioned earlier, the commonly held view was that all the root parts should have already been dead after 2 to 4 hours. Like long ago, it should be dead. But here we see this island still alive after 48 hours which is breaking records. The second reason, that I was amazed is that I'm a molecular biologist. I'm also a trained physiologist, but doing a lot of molecular biology, and we spend so much time on engineering and editing all kinds of genes, and rarely we see a phenotype. And usually, when we see a phenotype, it is a loss of function. We barely see any positive phenotypes. And here, without any engineering, without touching anything, we see a striking natural resistant phenotype. And apparently, what we thought, the 200 millimolar sodium concentrations of sodium chloride, like this is deadly. No, it's not. It's not lethal. It can be. We can overcome it. And this provides me a lot of hope that we actually can push it higher. Actually, we're not pushing anything. We are exposing the natural capacity of the plant, which is much. It can bear much more salinity than we think. Okay, so of course we repeated this result again and again, and we quantified it. And as you can see, after 48 hours and also 72 hours, 3 days in deadly concentration, the lateral roots, the top black line and also the green one, they remained alive, while the primary root, the main root in blue, was already almost completely dead. Interestingly, we found that the longer the roots are, they are behaving like a primary root, they're adopting a primary root nature, and they're losing their tolerance, as indicated by this red line of long lateral roots. And so here's a quick reminder of what lateral roots are and how they emerge. The lateral root anatomy is almost identical to the primary root, to the root tip. The exact same cell types we see in the primary root, we see also in the lateral root. The same, the same scarecrow. I chose scarecrow as the case as a presenter, but all the cell types are there. But for some reason, a scarecrow cell, if it is positioned here, it will die. But if it is positioned here, it will remain alive. And not only the scarecrow, but the whole island. Now, look, I don't want to make fun of myself, but I spent 5 years of my life studying Arabidopsis roots during little salinity. I spent hours on the microscope, and I was totally focused on the tip zone. And I never bothered to raise up my eyes in the microscope, like 1 millimeter up along the axis of the root, because I assumed, it's the root, it all behaves similar. But silly me, it's not the case. So, based on my own experience, my advice to you is don't get stuck in the root tip, raise your eyes, and be willing to explore out of the root regions. Okay, so let's continue. We repeated this result using other reporter lines. For example, this cell cycle regulator, a very important cell cycle regulator, cycling B1, which was fused to GAS reporter gene. And of course, the cell cytokinesis occurs in the meristem, so it's very active in the meristems. And during little salinity, so this is an entire root, it's a long one, lower magnification, the primary root there's no signal because the primary root is dead. But if we walk upper to the lateral root zone, we see beautiful signal of gas signal in the lateral roots because these cells the lateral roots are alive, they're active. Cyclin B1 is still active there, and indicating that in these meristems, that these meristems preserve some cell cycle abilities, which is very, very important for recovery because it is active, so we can now do cytokinesis and resume and produce new cells and regrow. Okay, so next we wanted to learn more about the regulation. Until now, I just described the phenomena, which was very surprising and exciting, and we wanted to know more about the regulation of it. So we measured reactive oxidant species, which are known to also to be involved in signaling and not only in stress in damage causing. So we therefore perform dab staining of the roots. Dab, it's a long chemical name, abbreviation. It is indicator of reactive oxidant species, specifically hydrogen peroxide. And it accumulates, it produces this brown color. And as you can see, again, we see an entire root. The primary root is mostly dead, but there's no signal there, but the lateral roots produce a lot of brown signal. They produce a lot of reactive oxidant species. Now, reactive oxidant species production can arise from multiple sources. One important source is the NADPH oxidase complex, which actively produce superoxide. It's very interesting complex, and so we suspected that this complex is responsible for this ROS production. The superoxide, like in a second, will turn into hydrogen peroxide, of course. And so we suspected this complex is responsible for the ROS induction, and therefore we treated it with DPI. It's the diphenyl iodonium chloride, which is a specific inhibitor which blocks the activity of this complex. And when we did so, we saw that the reactive oxidant species production was completely abolished. So we can filter out this ROS we see on the left. And then we, of course, wondered what this might do. Will this affect the tolerance, the salinity tolerance of these lateral roots? And the answer is in the next slide, in the bottom. So again, we see the same experiment. The primary root dies, but the lateral root remains alive. And again, we see another island, that the propidum iodide does not penetrate into the nuclei. And we see that the GFP signal persists. It looks beautiful. And when we treat this root with DPI, the lateral root dies. It loses the GFP signal, and the propidum iodide is penetrating into the nuclei and indicating that it is dead. So therefore we can conclude that this the ROS we observed in the lateral is actually a good one. It's not a bad one. It is necessary for, if you don't have it, you die. Okay, so this is actually a good one. And of course, the concept of good ROS is now well established. And there are many papers about this. Like this is a key reference, but there are many references now that support it. Finally, we wonder, excuse me for a second. Finally, we wondered what type of cell death is taking place in the roots during lethal salinity. So you probably know that there are several ways to die. Of course, at the cellular level, either through necrosis or through apoptosis. The problem is that plants don't experience apoptosis. Nevertheless, they have alternative cell processes, cell death processes, mainly these two, apoptosis like programmed cell death or autophagic mediated programmed cell death, PCD. So in the next two slides, I will show you that this is the case in little salinity in the roots. And let's see the evidence. So in case you didn't know, MDC, monodensyl cadaverine, is a specific indicator of autophagic endosomes. And so we did the MDC labeling, of the root during salinity, during deadly salinity, lethal salinity conditions, and we measured it in the two positions, like the main root as in the tip zone and in the lateral. And as you can see on the main, so let's check first the top panel. This is the primary root, close to the tip. You see all the cells have a lot of tiny bright green dots. Each bright green dot is an autophagosome. And the cells are full with these autophagosomes, which is not the case in the lateral root position, where the cells are absent. These green bright dots are absent from the cell's cytoplasm. To gain more information, more details about the structure of these tiny dots, we used more powerful imaging microscopy. We used transmitting electron microscopy. And in that case, you can see this is like the tip zone. And in the middle of the tip zone, there is a red box. And let's zoom into this red box. And we can get more resolution, higher resolution. And then we can zoom even more. And you can clearly see this beautiful autophagosome. You see multiple membranes because it is like engulfing several membranes inside with some cargo, perhaps some organelle. You can also see a phagophore. A phagophore is evolving phagosome, which is beginning to close itself on some randomly. This is assumed to be done randomly in the cytoplasm, closing up on some organelle. And then once it will completely engulf it, it will be turned into autophagosome, which eventually will be delivered into the central vacuole. And we can check, and we checked other cells within, the field I presented earlier, and we see these autophagosomes, and these phagosomes are classic marker of autophagosome-mediated PCD. There are few more signs for it, and we actually also determined them, but I'm presenting just this one. It is activated, it is found, these autophagosomes are found in the primary root, but not in the lateral root. Okay, so I'm now almost done. And before concluding my talk, I would like to make a brief summary. So this is a schematic, here is a schematic, this is a schematic seedling, a Arabidopsis seedling. And let's say this seedling is placed, it is situated in a soil which its salinity increases with depth. So the deeper we go, we find more and more salt. The higher the salt concentrations are. And such soils are actually very common in agricultural areas, as well as in some native natural habitats. So it's like not out of, out of nowhere. So this kind of environments can be very common. Now we need to make a decision, like the seedling needs to make a decision. Going deeper will expose the root to higher salinity and will make the stress worse. Therefore, it's better to avoid this direction, right? And on the other hand, it's better to promote sideways direction, which escapes the high salinity zone. We saw that the plant kills the primary root actively by autophagic mediated programmed cell death. We also saw that autophagic PCD is inhibited probably by NADPH oxidase activated ROS. We have more evidence for this. I can't go into details. I can provide it later separately. So we published several papers about this, and we think this is a novel adaptation mechanism that was not reported until now. As I said, apparently the lateral roots harbors higher resistance capacity, and that was unknown until recently. And so we published these papers, but of course we did not answer all the questions. Many questions are still unanswered. For example, is this phenomena, lateral root tolerance, unique for Arabidopsis, or can it be observed in other species too? Now, if this is common to other species too, then we can think about going, from the lab back to the field, and by simple nursery practices such as root trimming in the nursery, we can decapitate, the primary root, and by doing so, we will produce a sapling with more lateral roots, which supposedly will make the sapling more salt tolerant. And then, direct this kind of plants to salt affected lands that maybe these plants can be more suited to these kinds of soils. All these ideas were never tested. I didn't get the chance yet to test them. And there are many, many questions at the molecular basis and basic level and applied levels. And if this is interesting for you, so I'll be more than happy to, drop me an email. I'll be more happy to collaborate. And with this, I would like to thank again the organizer, Irene. Thank you so much. And thank the audience for you. Thank you very much for your attention. Thank you very much.
Thank you very much, Yori, for your presentation. Yes, many questions, but opportunities also for us to work in the future. So do you have any questions for both of the speakers? If you have any questions, please raise your hand and I will try to unmute you, or please write it on the chat. So let me check the chat. There is nothing. Okay, I can see one raised hand here. Let me try to unmute. Can you unmute yourself? No. Okay. Okay. Ali, can you hear us? I think you can speak now. Oh, you're mute again. Can you unmute yourself now? Because I have turned it off for you. No? Can you turn on your camera? Ali, can you hear us? Ali, you have to unmute yourself now. Yes, now you are unmuted. Can you hear us? Ali Volkan? I can see that you are unmuted. Can you hear us? I'm sorry about this, but I can see that you are unmuted and you can talk now. Okay, are there any other questions? Ali, can you write your question on the chat, please?
I see a question in the chat for
Okay.
Avi, for you.
So I can maybe read the question and then I can answer.
Yes, please.
You determined resistance varieties. Is it possible to apply for farmers? To answer this question, so the two varieties which we use, the first one, Salinity resistance variety, actually it's widely used nowadays in Philippines. So it's one of the salt tolerant variety which is already being used in different areas in agriculture. And another variety which we use for shade tolerant right now, this is not yet released because this is in progress. So we are working on it, that it's a in-house GWAS. We performed on that from International Rice Research Institute in Philippines, and now it's in the breeding pipeline. So I hope that answers your question.
Thank you very much. Other questions? Ali, can you talk now? You're still unmuted.
So I think the questions are written now, and Ali wrote it in the chat. So maybe I can read again the question. So it's towards me. So based on your results, do you see supplemental far red lighting economically feasible? So to answer this question, actually no, because so the supplemental far red which we used in the experiment in Wageningen University is to mimic the shade. And then in the field, how we make the shade is that we grow the plants together. But we have to keep one thing in mind, that it's not exactly the same, because supplemental far red is one of the cue of the shade, but then there are additional parameters. That's why we switched to the field experiment, where we wanted to understand how the plants perform, not just alone to supplemental far red, but other parameters, because there will be plant-to-plant interaction, volatiles. So with this study, we don't want to propose that supplemental far red is used as a commercially for rice cultivation, but the more we want to understand that if the plants are grown together, that how they perform in the field. And in the lab, we try to tease apart molecular mechanisms. So that's why we try to use the stimulus as clean as possible. Yeah, and then there is another question from Mehmet. How much EC decisiemens per square meter threshold for new rice varieties? So for this, so what we, the variety which I mentioned, RRI 147, which was salt tolerant variety, it performs fine in the mild salinity, so up to 8 decisiemens square meter. But right now, there are different breeding programs at Erie, which are focusing on going up to 12 decisiemens or even higher for coastal regions across Southeast Asia, for instance, in Vietnam. But there is a major effort being put there, but there is a very low success also, because to get a rice variety which is very pronounced or performing well in this high salinity is really hard to obtain. So, yeah.
Thank you very much, Pavinder. Any other questions?
So there is another question.
Yeah. Ali.
Sorry, yeah. There's another question from Ali. So what will happen in the case of prolonged far red light exposure in terms of seed quality and etc.? So that's a very interesting question. So right now, what we are doing is that this prolonged response, which is the shade response in this case, and supplemental far red is one of the cue. Right now, we are harvesting the plants and we will do seed quality checks. So we will perform elemental mapping. So like whole periodic table mapping for these seeds, to see what is the nutrient uptake and nutrient efficiency. And then we will also think about doing the grain quality. So right now I cannot answer that question, but hopefully in near future, I would be able to answer that. Yeah.
Perfect. Many thanks, Pavinder. Okay, let's see. I think if there are any other questions, I would like to thank you both once again. And our fourth Sustain webinar is about to end. So thank you both for your interesting presentations, and also all the members that joined this webinar. Thank you. Till next time. Thank you very much.